welcome back to another video on Arshbionyms. So in today's video, we are going to understand the mobility of the bacteria in semi-solid media. Okay. So in my earlier video, already we discussed bacterial motility, the methods to identify bacterial motility via flagellar staining method and hanging drop method. The videos on these topics are already available on my channel. You can check it out. In today's video, we are going to understand bacterial motility via semi-solid media. Okay. Now this is one of the easiest method to do and also the easiest method to read as well. Okay, now let us start with the video. Let us understand this particular concept now. Now first thing what we are going to do here is we are going to pick the organism on a sterile needle from the culture plate. So here we are going to make use of sterile needle and we are going to pick the organisms on the sterile needle. Alright. Now what we are going to do is we are going to take another test tube which will contain semi-solid media. So basically we are going to push the needle in the center of the tube about half a depth of the medium. Okay, so what we are doing, I have taken a test tube which contains semi-solid media. The same needle which is now having the inoculum, basically the organism on it. We are going to push this particular needle into the semi-solid media, but we are not just going to take it completely downwards. I am just going to take half a way into the test tube. Okay, we are not going to completely push it down, just half a way. Alright, into the semi-solid media. Now next is I'm going to incubate, and of course that after incubation the bacterial growth will be seen. है ना? Now if I want to know if the bacteria is motile or not, how to know it? So it's very simple. Either the result could be positive or negative. If the result is positive, that means if in case the organism is motile organism spread out into the medium from the site of inoculum. So you see, this was the area where I have incorporated my needle which was having the bacterial culture. So that is nothing but the area where my inoculum is present. So if the bacteria is motile, the whole bacteria will now move from the site of inoculum. Basically the site where we have pushed the needle, from that particular site it is going to spread all over into the test tube in this manner. Simple as that. So the whole test tube will now appear red in color. Okay. Now, what if the bacteria is non-motile? What if the result is negative? So, in case of negative result, the organism will remain only at the site of inoculation. So, what happens? So, if suppose this is the site of inoculation, that means that is the site or place where I incorporated the needle. Okay. So, the bacterial growth would be seen only near that particular area. So, only bacterial growth would be seen only at the site of inoculation. They will not spread. Simple as that. So positive result मतलब क्या होगा? What happens in positive result? The whole test tube, the bacterial growth will be seen in the whole test tube because the bacteria is motile and moving in every corner of the test tube. If the bacteria is non-motile, we cannot see the growth in the whole test tube, but only where we have inserted the needle. Because they are non-motile, they'll grow only wherever we have pushed the needle inside. I've incorporated the image here. You can check it out. So if in case you see this particular test tube which is completely red in color, this indicates that the result is positive and the bacteria is completely motile and moving from the site of inoculation to the rest of the test tube. Now in the other test tube, if you notice, this represents the bacteria is non-motile and the growth is seen only wherever the needle was pushed inside the test tube, only at the site of inoculation. So this is how we perform this method. So with this, all the three methods to identify bacterial motility is over. So if you combine all these videos, it's going to become your essay answer, which is available on my YouTube channel. And I hope the class was understandable to all you guys. So let's meet in the next video.